Greetings everyone, and today we are going to discuss just shortly about significance of bacteriophage in genetic engineering. First, let's discuss what is the genetic engineering. Genetic engineering is a field in which we study about the alteration and modification of genes in order to obtain a desirable product for the welfare of mankind. Now, here, genetic engineering first and foremost includes a lot of factors. Some common terminologies in genetic engineering includes gene of interest. So gene of interest is a gene. For suppose uh, if I want a product, a genetically modified product, which is known as insulin. Let me suppose if I'm having insulin, if I need insulin. Now for insulin, the gene which can make insulin within the human body will be taken from the gene, the gene which makes insulin will be taken from the human body cell, as a result of which that gene which will be taken from the human body can be utilized in another organism such as bacteria or bacteriophage to produce a desirable product which is insulin. At that moment, this gene which will be producing my desirable product or insulin will be known as the gene of interest. That gene of interest is responsible for giving me my desirable product which is insulin, which I want in bulk quantity. Now, how does this process all work? Let us discuss. Let us suppose we are having a gene of interest. Now, the gene of interest which I have taken from an organism will be taken by the help of an enzyme known as molecular scissor, commonly known as molecular scissor enzyme, which is restriction endonuclease, which basically causes to cut the genome of the cell. As a result, the particular genetic material, particular gene genome from the cell, and that genome is then that gene of interest is taken from the genome. And at that moment, that gene of interest is then inserted into another organism, and that organism which would be providing us our desired product, in which we are going to insert the gene of interest and which is going to give us the product insulin, is known as the genetically modified organism, GMO, commonly known as GMO. And that GMO will have the gene of interest inserted inside it. Mostly we take the genetically modified organism as bacteria and bacteriophage. Out of bacteria and bacteriophage, we mostly take bacteriophage. Uh, not no, sorry. A genetically modified organism. There are two organisms, bacteria and bacteriophage. What we really, why do we uh, take bacteria as a major organism? We mostly take bacteria as a major organism within it, because bacteria is first and foremost prokaryote. It is having the simplest structure, as well as it is having the highest reproduction rate, which means within 20 minutes, within approximately 18 to 20 minutes, it within 18 to 20 minutes, it creates two molecules of insulin, two molecules of insulin. So how fast this rate is now, prokaryotic in prokaryotic cell, when it is injected, first and foremost it is having simpler structure, so it is easy to inject, as well as it is having high rate of reproduction, as well as it is also having a circular DNA, due to which it is easy to insert it within bacteria. Now whatever the genetically modified organism it is, the genetically modified organism, when will replicate itself, it will cause to replicate uh, the gene of interest as well. For suppose this is GMO, this is a GMO I've taken, and this GMO has the gene of interest I've inserted. Let's suppose we are having insulin in it, insulin gene of interest in it. Now when it will replicate, it will form several other GMO, and when these GMO will replicate, they will further replicate into other GMO, and when these GMO will replicate, they will further form multiple copies of such GMO, as a result of which, as GMO will start to replicate, the gene of interest will start to replicate, And as gene of interest will start to replicate, more of the desirable products would be made from multiple GMOs, as a result of which our product will be obtained. So in such manner, this is the basics of genetic engineering. Now when we are inserting the insulin, a gene of interest, within the genetically modified organism, the same enzyme restriction endonuclease is utilized, which is inserted within the which causes to cut down the genome of the genetically modified organism. And after it is cut down, what we actually do is take the gene of interest and insert the gene of interest within the genetically modified organism GMO's genome, as a result of which it causes uh, to form a genetically modified organism's genome, and that moment it further undergoes replication and produces our desirable product. Now bacteriophages are having such importance. Bacteriophages first and foremost are utilized in formation of several genomic libraries. Now what are genomic libraries? Genomic libraries are basically those libraries of bacteria and bacteriophages which contain multiple copies of different types of genes of a living organism. Let's suppose if there is a genomic library of human human cell. If there's a genomic library of humans, then it will include multiple copies of the genes present within the bacteria and bacteriophage. It will include several types of, let's take bacteria and bacteriophage, which will have several copies of different types of genes of the human cell proteins, as a result of which these genomic libraries are further utilized for making such products, among which example is insulin, and other types of products can also be obtained. Moreover, there is another application which is known as phage therapy. Phage therapy is a therapy in which we directly utilize bacteriophage as a sort of medicine, because bacteriophage are very specific in their nature. They attack only a specific type of bacteria, as a result of which, as they attack only a specific type of bacteria, they can be utilized and encoded within their genome to attack a particular type of bacteria within our body, so that we can easily get rid of bacterial infections without damaging or having our floral bacteria present within our body to be harmed, as a result of which this can also be utilized in phage therapy. Another advantage which I would like to highlight is a recent one, which is known as antibacterial nano medicine. It is also utilized, bacteriophage is also utilized as antibacterial nanomedicine, which means that as antibacterial nanomedicines, many of the medicines, uh, many of the diseases, bacterial diseases which are not easily treated by uh, medicines, antibacterial nanomedicines contain bacteriophages which work against a particular type of uh, bacteria in order to kill them and get rid of dangerous diseases such as tuberculosis and other such diseases. So this is just a short intro and significance of bacteriophage, and that's all about this video. Hopefully you enjoyed the video, and hopefully we'll meet again in another one. Till then, you can like the video and subscribe to it. Thank you.